Viruses have to infect a host, so viruses are not able to have reproduction on their own like you or I can do. So instead, what a virus does is it infects a host, and then it has to rely on the host to replicate for it. So each virus is tied to a specific host range, and the host range of a virus the host range of a virus is the specific species that have cells that virus can infect. Most host ranges are very small, so a lot of viruses have only one species of host that they can infect. Influenza is an example of one that actually has a larger host, and the different strains of influenza are going to infect different hosts. So you can see influenza A, B, C, D in the picture. And in the center, you can see men and pigs, which are susceptible to all 4 different strains. When you hear terms for the flu, like swine flu or the bird flu, often that's talking about the original host for that strain of the virus. So the swine flu in 2009 was actually a strain of flu found in pigs, and enough mutations happened in pigs that some of that virus was able to jump hosts to people, and then we called it, swine flu or H1N1. We've heard bird flu. There's a lot of influenza A viruses that affect fowl, and then influenza D tends to be more of like these ungulates, cattle, goats. So the host is going to depend on the specific genetics of the virus and what it's evolved to infect. And when a host jump happens, that's when we start to see like a new virus appearing in man. It's not a new virus. What happened is it was in a different type of species, and it jumped hosts through mutations or because of new contact with the species. This is what happened with the HIV virus. So HIV originally infected chimpanzees, and then a host jump occurred from chimpanzees to men, and then humans spread HIV. So when a virus replicates, it's going to go through 2 different cycles. There's 2 possibilities, lytic and lysogenic. And in these 2 different cycles, there are some general similarities. So in order for a virus to reproduce, the very first thing it has to do is it has to enter a cell. It can't just float around in the blood. A virus has to actually physically enter a cell in order to get to the manufacturing proteins. So once the virus enters the cell, it's going to use the viral DNA to start making viral proteins. And it's going to use the host to do this. So it uses the enzymes from the host, the ribosomes from the host, tRNAs, amino acid, ATP, and energy, all other molecules coming from the host, right? So viruses only bring with itself the proteins that it needs to have kind of a structure and to be able to infect cells, right? Like the spike proteins on the outside help the virus identify cells and infect them. And then it brings its instructions, its DNA. Everything else, all of the tools are part of the host cell. And then eventually the virus, once it's made enough proteins and DNA to reassemble itself, will reassemble itself into a new virus. And this might happen very quickly, like the lytic cycle, and it might take years if the virus spends some time in the lysogenic cycle. So let's go through the lytic cycle first. This word lytic has the same root word as lys, right? So lytic means to break or burst. The lytic cycle is typically a very fast reproductive cycle. So this is where it's producing new phages, right? Phages are my virus. And then it's bursting open the host cells and releasing all of the virus progeny, the babies. They're not babies, they're new viruses, out into the blood to go infect new cells. And then each new cell gets infected, makes a whole bunch of viruses. They break open, release those viruses. So this is going to increase the amount of viruses in the blood exponentially, very quickly. So a virus that only goes through the lytic cycle is typically a very virulent phase. Virulent meaning that they're going to be very disease-causing. They're going to have a quick onset, not a super long dormant period, and they're going to kind of magnify the symptoms in the host very quickly. All right, so step 1 is attachment. Here's a bacteriophage, and this is my bacterial cell. The first thing it's going to do is it has to identify the cell. And this happens in all of the different types of viruses, like COVID-19. The spike proteins on the outside of COVID-19 identify the ACE2 receptors in our cells, and that's how they attach. This is a bacteriophage, so it's going to attach down here with its spikes. And then the DNA is going to enter into the host cell. So in this case, we're doing the lytic cycle, and this is going to degrade the host DNA right away. So this bacterial cell no longer has its own instructions anymore. All it's going to have now is the DNA from this new virus. So the cell's going to use the DNA instructions from the virus to start making new viruses. It's going to make the proteins for it, makes new copies of its DNA, and those self-assemble. The self-assembly is really cool. The self-assembly, the virus doesn't need any kind of host help with the self-assembly. Over time, the virus parts organize themselves into the correct shape spontaneously. So I could self-assemble a virus. Like I could take all of the virus parts, put them in a cup, and shake it up. And if you just put a little bit of energy or even just time into it, they would self-assemble themselves into the correct shape. And then they burst out of the cell. So this is the last step of the lytic cycle, is the breaking. So we started with one virus particle. And then now my bacterial cell is dead, because when you break the cell membrane, the cell is going to die. And we have 5 new virus particles. So imagine this starts at 1, it goes to 5, 25, 125, and that's only 3 cycles to go from 1 to over 100. So as this reproductive cycle occurs, the number of viruses is going to increase dramatically. The lysogenic is a little slower. So the lysogenic cycle will eventually go into the lytic cycle, but it's not going to happen immediately. So it does not destroy its host right away. So what happens is the virus gets integrated into the DNA of the host. Often this is going to have a piece of virus that stays in the host DNA for a long time. And this is called a prophage. So prophage meaning that pro before and phage being the virus. So it's a piece of virus DNA. It's not in a virus yet. It's still in the host, but it has the potential to become a virus. These are typically more temperate phages. So you're going to see viruses that kind of have a slower onset, have a longer kind of dormancy period. So something like HIV enters the body through human contact, but you don't immediately get AIDS, right? HIV can kind of be dormant in the body for a very long period of time, and then at some point it will go into the lytic cycle, and that's when you start to see symptoms of AIDS start to occur. And the longer the virus stays in the lysogenic cycle, the longer you have before you start to see kind of symptoms. So for SARS-CoV-2, which causes COVID-19, we believe it stays in the lysogenic cycle for at least a couple of days, which would explain kind of how it takes a week for symptoms to onset. But there are reports that it can stay in the lysogenic cycle for up to 14 days, which is why sometimes you see people who get infected but then don't have symptoms onset until 2 to 3 weeks later. All right, so let's follow the pathway of the lysogenic cycle. So we're going to start up here. Our starting point is right here, where the phage is going to infect the DNA into the host cell. If it's lysogenic, it may or may not destroy the host cell DNA right away. So in this case, we are going to see, here's the virus DNA, and this is a single-stranded circular DNA, and then here's the host DNA over here. And so it is not destroying it. Instead, it's going to enter the lysogenic cycle and make a prophage. So the piece of virus DNA actually gets inserted into the host DNA. This is something that happens in herpes in humans. So people who've had herpes can get outbreaks of herpes on their genitalia, on their face, wherever the herpes virus is. And what happens is the herpes virus is always there. It's always in the cell. And then during times of stress, that's what causes an outbreak. During an outbreak, some of those cells will go into the lytic virus, and you'll start to see the symptoms, which are like the genital warts or warts on like the cold sores on the lips. But for the most part, the herpes virus stays in this kind of dormant prophage system. And during this time, so this is a bacterial cell, this bacterial cell is going to replicate. So when it replicates, it's going to make 2 new cells, right? One here, over here. And in each of these cells, we just made a copy of that virus DNA. Here's the virus DNA. We made a copy. And so the bacteria are going to replicate over and over and over again and make many, many, many copies of this viral DNA until you have an entire culture of bacteria with the virus DNA inside of the genome. Eventually, at some point, it's going to switch into the lytic cycle, where that virus DNA will come out of the host DNA and then start into the lytic cycle. So if a virus stayed in lysogenic forever, we would never actually get symptoms. So the actual virus symptoms that are caused happen when it switches to lysogenic. And the amount of time switches to, so the actual symptoms occur when we switch to the lytic cycle. And the amount of time it takes until symptoms start depends on how long a virus stays in the lysogenic cycle. So something like herpes will stay in the lysogenic cycle almost all of your life with minor outbreaks, right? At different times, you might see some cells switch to lytic and then the outbreak goes away. Something like HIV might remain dormant in the cells for a very long time, and then eventually something might switch it to lytic, where you start to see massive changes in symptomology, people who start to develop AIDS. In COVID-19, so SARS-CoV-2, which causes COVID-19, the lysogenic cycle might last anywhere from as short as 3 days to as long as 14 days. And then eventually it switches to lytic, and that's when you start to see like the fever and the coughing and all of the symptoms of COVID-19 occur. Bacteria have some really cool defenses against viruses. So now that we've seen how the virus invades the cell, we can see how we can fight against some viruses. And bacteria have they have proteins called restriction enzymes. And restriction enzymes cut DNA. So if the virus infects me with its DNA, I can take my restriction enzyme and chop it up. And now the DNA can't do anything to me because it has been cut. Another thing that can happen is the bacteria protects its own DNA by being methylated. So it's got restriction enzymes that cut up foreign DNA, but those restriction enzymes can't cut up its own DNA. Bacteria also have something called the CRISPR-Cas9 system, which can target specific sequences and then cut them. So this is another mechanism for cutting up DNA. We've been using CRISPR-Cas9 to kind of make targeted changes while we study DNA. But bacteria can use this to target specific sequences in viral genomes, cut them up, and help protect themselves against those viruses. So what we end up seeing is an evolutionary kind of co-relationship where the bacterial cells are going to be at an advantage if they have things like restriction enzymes, CRISPR-Cas9, that can cut up viral DNA and they're protecting themselves. So those genes are going to get passed on. And then viruses, in response to that, will be at an advantage if they can make mutations that make them more effective or change their DNA so that it's not recognized by the restriction enzymes. And then those will be at an advantage. And then the bacterial cells can develop new restriction enzymes or new CRISPR-Cas9 sequences to protect themselves against these new viruses. And so we see this is kind of why the host ranges of most viruses are extremely narrow, where a bacterial phage will only infect a specific species of bacteria because they've co-evolved together for so long that the virus would be virtually ineffective if it infected any other organism, except for the one specific organism that its sequences can infect.